Hi, everyone. Welcome to Exploring Science. Today's exploration. The first talk will be from Dan Konstantinovsky, and he's a graduate student in biophysics and he will be teaching us about molecular symmetry and what happens when it disappears. So he can probably answer more questions about this from our take-home activity that we just went through. And our second talk will be from Victor Olmos, and he's a graduate student in genetics, and he'll be giving a talk on his family's neurological disease and how he started his journey to curing it or trying to cure it. So with that, I'm going to hand it over to Dan, sorry. And Dan, if you want to share the screen.
Actually, Victor is going to go first.
Oh, is it Victor
we changed the order.
Hi, everybody. My name is Victor Olmos. I'm a first year here at Yale, and I'm very excited to give you my talk about inheriting death. And the very beginning of this video, there's going to be a video, so I recommend increasing your volume for it. It's a little quiet, and then you can lower your volume after the video is over. So let's begin.
I think about hunting. It's an writing movement, and I hate
Oh, I'm sorry.
Seeing her, just a shadow of what she used to be, is not as I want to remember it. Kimberly met Justin just before she did her OEs, and she said to us, I've met someone. And about a week before she got back, he arrived in one night and he said, I've got something to ask you both. And he asked us, could he marry her? I told him about Huntingtons. I told him all the worst things about it, and he said he was fine with all of that. He just said, no, I still want to marry her.
So the disease the person in the video had, it's called Huntington's disease. And what this disease is it's a dominantly inherited disease. What that means is if you have two copies of every gene in your body, if you have just one copy of that gene, you have the disease and you will show it. Another thing is it's a progressive neurodegenerative disease, meaning that it shows symptoms usually about how the symptoms don't occur till later on in life, usually your 30s or 50s, and it slowly occurs over time. Similar to diseases like Parkinson's or Alzheimer's, it slowly occurs. You don't just be born with it and it happens over time. In the video you saw, it causes a loss of movement and thinking. The person, she can't control her body. You jitter a lot, you seize a lot. And what is it caused by? Well, it's caused by something called the CAG expansion. And what that is you can see here, there's these DNA bases on top, and DNA is made of A, T, C, and G. And when you get a bunch of these CAGs in a row, it makes something called glutamine, an amino acid. And amino acids make your proteins. And when you get a lot of these glutamines in a row by these CAGs in a row, you make a lot of glutamines in a row. And that's what causes the disease, this expansion of glutamines. You can see here that humans normally have 6 to 36 of these. We have a lot of them in a row. But when you get more than 36 of these repeats, you have the disease. And a fun fact that glutamine, it's abbreviated as Q. I don't know who decided to abbreviate a G word as a Q. You know, an A word got abbreviated as an A, but a G got abbreviated as a Q. Some scientists out there just had fun with it. But because of that, we call these diseases Polycube diseases. Poly means many, Q, glutamine, many glutamines, Polycube. So this disease, what it does is it really affects the brain. It affects the medium spine striatal neurons. I didn't make the name of the neurons, it's just the name of them, and the basal ganglia region. Here I have a picture for you of a brain, a Huntington's brain and a healthy brain. Can somebody guess which brain is the healthy brain? Left or right? Which one's healthy? Which one do you think? Yep, the right, correct. And why? Well, because the brain on the left has a giant hole in it. You know, there's a lot of brain missing on the one on the left. And so what region's affected? It's the basal ganglia right there. And that region plays a large role in movement and behavior control. Interestingly, though, Huntington is made in all cells in the body and the brain. So why is it only that region right there is affected and not the rest of the brain? Why is only a part of the brain being affected? Well, we don't know because we don't know what this protein even does normally. We know what happens when you have a mistake in it, but we don't know what this protein does normally in the body. So very hard questions for us to answer when we don't know what this thing does anyways. We do know, however, some features of the disease. The protein, with those expanded cues in it, it will fold improperly, and it makes clumps. And these clumps clump together. And you can think of the disease as a snowball. Now, if you get hit by a snowball, it hurts. But that snowball, if you put it on top of a hill and it rolls down the hill and gets more snow, more snow, more snow, it becomes a big snowball. If you get hit by that big snowball, it's going to hurt a lot more. So you can imagine that in the cell. These snowballs form and they grow over time. It's progressive disease. And by the time you're in your 30s and 50s, it's getting bigger and bigger until eventually it will kill you. I see a question of why does it clump together? Those cubes, those glutamines that form together, it just folds incorrectly, and they bond each other through different mechanisms, and the body will send the congregate together. So, yeah, that we don't know the answer of that entirely. But why am I sharing this with you? Why am I talking about Huntington's disease? Well, the reason is my family has this disease. These are pictures of my grandfather, and he inherited the disease, and he suffered and he died from it about a year ago now. And it's what drove me from a very young age, about 10 years old, to go into science and wanting to go into a PhD program to find a cure for this disease. And I luckily didn't inherit this disease, but my dad has 8 sisters and 2 brothers. Many of my family members have this disease and will die from it. And I keep talking about how do you inherit this disease? So I want to go over how you map it out. These are something called the Punnett square that some of you may or may not have seen already. And what you do is you have on one side, one parent, an example here is the father that has a disease, on the other, the mother. Now the Huntington gene is this big red H I did for you. And what you do is you just fill these boxes going down and across, and you fill them out. Each box is a 25% chance that that offspring has a disease. So if you just fill the box out, so the top one, you have 1 copy of the disease, you have a disease. If we keep filling out this box, can anybody tell me what percent of people will have the disease? What percent of children will have the disease? Exactly. Half of the people will have a disease with one parent. The disease is rare, so we usually assume that half will have it. But now let's see if both parents have the disease. What would the odds be? Do you think it's going to go up or down? Yep, it's going to go up. Now let's just fill the boxes out. Here we have 2 copies, 1 copy, 1 copy, and no copies. So if you had both parents that had the disease, 75%, 3 out of 4 kids, offspring, will have the disease. And interestingly, in the case where if they have both these copies, they might not even survive being born. It's that severe. Now you can also map this out in something called like a family, like a pedigree chart. Here is a snippet, a small portion of my family. I have 8 aunts, 2 uncles. Here's only showing like 4 of them. This black circle is my grandfather. These circles and squares are my aunts and uncles. But luckily, this was my father right here who didn't have it. So I do not have it. But just because I didn't have it didn't mean I should just live my life, ignoring it. I'm trying to work on something very similar to this. And so there are 9 known Polycube diseases out there. There's, again, a reminder, Polycube, the glutamine, it's a Q. And the 9 are, I can't say this word. I'm a bio major, not an English major. So we just call it DRPLA. The other one is Huntington's disease, spinobulbar muscular atrophy, SBMA. And there are 6 spinocerebral ataxias called SCAs. And here at Yale, I'm working on SCA1. Now, you may be wondering, why am I working on SCA1 and not Huntington's disease? My family has Huntington's. Why am I working on it? Well, I don't have my PhD yet. I'm still a very young scientist, and I don't feel like I'm ready yet to work on that, both, at the science level and emotionally. I'm still not there yet. So I'm working on a disease called SCA1 that is very, very similar. Why did it skip to 17? Well, there's many SCAs out there. There's like 40 different SCAs out there, and only we numbered them by when they were discovered. And so it went 1, 2, 3. We skipped a few, 6, 7. We didn't know about a couple, and that's 17. The naming's weird. I'm sorry. But again, going back to my disease SCA1, what it is very, very similar to Huntington's. I'm going to share that with you right now. So I made this chart. Green means that's identical, yellow very similar, and red very different. So going through the chart, you can see SCA1 dominantly inherited, progressive disease. Age of onset is about 10 years more, so sooner than Huntington's. It's, again, caused by a CAG repeat, but in a different protein. This one is also, you can have up to 39 repeats instead of 36. However, we do know the role of ataxin-1. So we do know the role of the protein. So it's easier than Huntington's, in that we can understand when it's messed up, why things were messed up. It's made in all cells in the body. But again, this disease now is in different cells. Huntington was in the medium spiny neurons and the basal ganglia. SCA1 is in the cerebellum. And where the cerebellum is located, here I have an MRI scan. In the circle right here is a healthy cerebellum. And in this other circle down here, you can see a diseased cerebellum. And you can see it's much darker. There's much less cells there. Now on the right, you have two mouse images of these cells in the cerebellum. Can anybody guess which one has the disease, top or bottom? Exactly, the bottom. And you can see that there's a lot of cells missing, and it has 82 Qs. 82, much larger than 39. They have the disease and these cells die. Now, luckily we can use mice as a model organism for the disease, so we're not just using humans. We have mice. This mouse, for example, has 154 Qs, more than 100 Qs greater than the disease. This image right here shows colorful dots of where the protein's found in the brain. As I said before, the cerebellum, this yellow area back here, is affected and the cells die, but it's expressed everywhere in the brain. So why isn't everywhere else in the brain affected? We don't know. Here I have a timeline of a mouse. At 30 weeks, the Purkinje cells, they start to die. But if you look to the left, you see that at 5 weeks, they can't walk. 6 weeks, the snowball starts to form and clump together. 7, 8, they can't think correctly. 11, they have weight loss. So how are all these things happening before these neurons die? We thought before it was neurons dying that led to the disease. Now we're thinking that other things are happening that lead to these neurons to die. Now, this disease, SCA1, and the other Polycube diseases, we do not have a cure. We barely have treatments for them. Once you have them, you will die. So how do you cure these diseases? What am I trying to do to work on it? Well, I made a 3-step plan for you guys. The first step would be to stop the forward progression. What I mean by that is, here's a simple timeline from birth to natural death with different parts of the disease occurring, where they can't walk, they can't talk, think, and then eventually dying. Now, what if we made it such that they don't die early? The person still can't walk, talk, or think, but now they don't die. But what if we catch it earlier? We catch it before they even have symptoms. And then we prevent them from ever losing the ability to walk, talk, or think. They don't lose that. So they can live a healthy life if we stop the disease from occurring. Now, step 2. Well, what if we get somebody that already can't walk or talk? You know, we could stop the forward progression where they don't die early and they don't lose their ability to think, but they can't walk and talk anymore. So how do we go from not being able to talk and walk back to a healthy person? How to regenerate those pathways, those neurons? We don't know. We're working on that. And finally, the third step, how do you stop the transmission of the disease? How do you stop it? How do you make it that you never inherit death? How do you go from having this possible timeline to this timeline, to just living a healthy, normal life? How do you get these family pedigrees and get rid of them? We don't know. And science is going to it takes a very long time sometimes. I've been wanting to cure this since I was 14 years ago. It takes a long time, and I might not cure it in my lifetime. Maybe one of you will go into science and work on this field, hopefully. And maybe one of you will help cure my family and other families like mine that are out there. And I just wanted to give one big shout out to my lab. I miss them dearly. I haven't seen them in a while. You know, we used to go hang out and get food together. I love sushi, so I just wanted to put a sushi picture in there. And just thank you all for listening to my talk. And if you have any questions, I believe, yeah, thank you.
Okay, thank you so much for a great talk, Victor. I'm going to ask a few of the collected questions. So I think, you did a great job of answering questions actually during the talk. But maybe about these Polycube diseases, do scientists think that there are more out there? Have we discovered any like really recently?
We haven't discovered any for a while, actually. Thanks to advances in genetic screenings, we can screen people. And so if they have a disease that affects the way they move, they can analyze their DNA to try to find these. SCA1 was discovered back in 1993. Same with Huntington's. So these were discovered 26, 27 years ago. We found out that the polycube was affecting them. And so for the past 27 years, we've been working on finding out how they work and trying to find a cure, but we still haven't gotten much done.
Okay. Let's see. If I guess sort of an ethical, maybe a science ethical question. Why, if you could stop the disease at the later stage, like before death, but after someone was already completely immobilized,
yeah.
Is that what? It's difficult to phrase this question, but I guess how do scientists think about the quality of life that such a person would experience?
I mean, that would be up to the person if they wanted that treatment. If they thought, they can't walk, talk, or think, do they still want to live forever like that, or they rather die? That is a question on the patient rather than the scientist. We just provide the opportunity that if we get someone earlier on, that they'd never have the symptoms. So it's up to that person and their judgment what they kind of want life they want to live. Maybe they want to prolong their life and then hope that step 2 comes and they regain those pathways and then can talk again and think again. It's not my job to decide that for them. It's my job to find cures and treatments for them, to give them the option and the opportunity to have that chance to live a healthy life.
Okay, and then finally, about the disease itself, what is the a difference that causes a person to die before even being born? Is that the number of clumps or? Yeah,
so like I showed before, when you had one H, you have the disease at 30 to 50 years of age. It starts more or less. But when you have 2 of them, you had a healthy copy and it was doing normal stuff in the body. But when both of them are messed up, for Huntington's anyways, they die when they're born because that copy, we don't know what it does, but we know you need it to be born. So for Huntington's, you need to have one healthy copy or else you can't even survive birth. For SCA1, you die around 4 years if you have both of those copies. So they're needed for development and growing. How exactly? We're still working on that. And so having one healthy copy, you can live for a little bit before the other unhealthy copy affects you all the way. So that's why both copies is way more lethal.
Okay. Well, thank you so much, Victor. I think there are still more questions, but we might get to those after the talks.
Yep.
And I'm sure everyone really appreciates you sharing such a kind of a personal story. And it was very inspiring to me, and I imagine to many people in the audience.
Thank you.
Our next talk is going to be from Dan Konstantinovsky. And he's going to be telling us about symmetry in nature.
Hi, I'm Dan. I'm a grad student. And today I'm going to talk about symmetry in chemistry and biology, but especially I'm going to focus on what happens when chemicals have no symmetry at all. Turns out that's very important for chemistry and even more important in biology in ways that we will see. So it's good that you had this symmetry exercise before this, because I think now you're sort of all prepared for this kind of thinking. But just to go over symmetry briefly, let me just go over 2 common kinds of symmetry. So let's say that we have a water molecule where the white spheres represent hydrogens, the red sphere represents the oxygen, and the connections represent the bonds between them. So this molecule has what's called reflection symmetry, which means you can draw a line, for example, through the middle of the molecule, and then take one side, for example, the right side, and then reflect it onto the left, and you get the same thing as what you have on the left. That's reflection symmetry. There's also such a thing as radial or rotational symmetry. So for example, if you look at a water molecule from the top, and then you twist the water molecule 180 degrees, so half a circle, you see that even though the hydrogens have switched places, the molecule looks the same. So that's called rotational symmetry. And generally, most molecules that you think of have some kind of symmetry. But what about this molecule? So it doesn't matter what exactly each sphere represents. Imagine that there's just 5 different atoms bonded together, and they're all different. So if you think about it, you'll realize that there's nowhere that you can draw a line, or there's no way that you can turn this molecule to make it look the same. There is no symmetry in this molecule because every single sphere is different. So now I'm going to ask you another question. What do you think about this molecule? So what I want to ask you is, do you think that these two molecules are actually the same molecule, just seen from different perspectives, or are they actually different? I'm going to give you a couple of seconds to answer that. The correct answer is actually that they are different. And you can see that if you were to, for example, take the molecule on the right and twist it around such that the purple sphere and the red sphere are aligned. So you're trying to align as much as you can. So let's do it now. So that's what happens when you take the molecule on the right and you put it on the left. You see that the red sphere and the purple sphere are aligned, but the green sphere and the white sphere are now out of order. So as you can now see, these two molecules are actually different. But how can that be? How can two molecules that look exactly the same actually be different? So before I continue, I'd like everyone to hold their hands out in front of them and think about your hands. Now, take your right hand and try to twist it around in such a way that it becomes identical to your left hand, and then put it on top of your left hand. So do you see how even though your hands are the same, they look the same, there's no way you can really turn a right hand into a left hand. So they're actually different. And it turns out that there are molecules like the one that I just showed you that are different in the same kind of way. So this property of molecules was discovered by Louis Pasteur, a French scientist, when he was looking at the products of what happens if you take wine and you boil it for too long, you get this solid stuff at the bottom. And he was looking at the crystals at the bottom of the wine, and he looked really close at them. And he discovered that there were two types of crystals, one that looked like the one on the left and one that looked like the one on the right. And there was nothing he could do, no matter how much he twisted the crystal on the right, that could make it look like the one on the left, even though they looked the same. So he realized that they were exactly like your two hands. And basically they had handedness. And these are the molecules that actually form these crystals. And you can see that they have the same handedness property that molecule that I showed you before had, and the same thing that your hands actually have. So Pasteur called this phenomenon chirality because it comes from the Greek word cheri, which means hand. So handedness and chirality is the same thing. It's the property when you have no symmetry and when you can have two molecules that are mirror images of each other, but cannot be turned into each other. Okay, so why is this important? It's really cool, but why is it important? Well, let me give you a really interesting example. In the 1950s, scientists came up with a drug called thalidomide. And thalidomide was one of those molecules. It's got a left-hand version and a right-hand version. So it turns out that the left-hand version in this picture helps pregnant women with morning sickness. It prevents things like nausea and other symptoms in the early stages of pregnancy. But the molecule on the right-hand version of the same molecule causes really horrible birth defects like missing limbs, like missing arms, fingers, legs, and potentially even worse. And there were many, many babies born with these kinds of deformities because of thalidomide. And the problem was when this molecule was being made, both types of molecules, the right and the left-hand part, were made in the same amount, 50-50. So when someone swallowed a pill, it was half like the orange molecules and half like the blue one. So that obviously caused a huge problem. So then, okay, that's obviously important. But then why do molecules with different handedness, drug molecules, have such a different effect on the body? The reason that the body cares about the handedness of a molecule so much that one can cause birth defects and the other doesn't is because the body itself is full of molecules that have handedness. And if you think about it, think of the molecule of DNA. Molecule of DNA, you probably have seen it. It's like a double helix, like a ladder. And if you follow this molecule, you can see that if you take your right hand and you twist it inwards and you go up at the same time, that's how the helix twists. The ladder goes up like that, right? You could also imagine another type of DNA molecule where everything is the opposite. Instead, your left hand does the same thing. These are not actually the same, remember, because they have different handedness. The interesting thing is that if you look at the DNA in our own cells, only the right hand version is actually there. So you realize that basically different molecules in the body have this different handedness. And because they have different handedness, they interact differently with drugs that have different handedness. Kind of like it's easier for a right hand to shake a right hand than it is for a right hand to shake a left hand. And I'll get back to that later. Oh, so artificial DNA facing the other way has been created, but they haven't created any organisms with that kind of artificial DNA. Although you could imagine doing that, and that would be super cool. And there are people who do that. So, okay, again, now I've answered the question. So why drug molecules might interact differently with different handed molecules in your body. Okay, but then where did this handedness come from? Why is all of our DNA right-handed and not left-handed? So this question is a really, really hard question to answer. But I'm going to try to give you some clues. So imagine the early Earth, the origin of life, 4.5 billion years ago. Very different world, much hotter, much more volcanically active, all sorts of differences. And you have these rocks and you have these pools. And in these pools, chemical reactions are happening. And these chemical reactions were thought to involve molecules very similar to DNA. The special thing about DNA and related molecules is that DNA is able to make more of itself by a process very similar to a handshake, and by basically templating another molecule. So you can imagine one molecule of DNA assembles another one basically by shaking hands with this other molecule. And now they have 2 molecules of DNA, and then they can make 4 and then 8, etc. So if you think about this process being like a handshake, think about it this way. Imagine that for some reason in the beginning, there was slightly more of the right-handed version of DNA than the left-handed version. And the right-handed version preferred to shake hands with other right-handed versions to make more DNA. And then that just kept going and going and going until all the DNA was right-handed. So something I'd like everyone to do is try to shake your own hand. Try, for example, putting your right hand into your left hand. Do you notice how awkward it is and how different it is from when you shake hands with someone and you're using your right hand? Well, the reason is because your hands have opposite handedness and they don't fit. And so the same thing, if there was a little bit more of the right-handed DNA, it would prefer to make more and more and more and more right-handed DNA. So that kind of answers the question. So, but then, okay, let's really answer this question. So in the 1950s, 2 scientists named Miller and Urey performed an experiment where they took a bunch of really simple molecules, simple chemicals that they thought were present on the early Earth, and then put them into a chamber where they simulated the conditions of the early Earth. For example, the early Earth had a lot of lightning, so they added artificial lightning and other conditions. And they wanted to see, could biological molecules like DNA form spontaneously? And the answer is, some of them actually did form, which is pretty cool. However, unfortunately, the handedness of these molecules was about 50-50, right-handed and left-handed. So that means that scientists have an explanation for maybe how these biological molecules can arise spontaneously, but we don't really have a very good explanation for why there would be more of the right-handed version or the left-handed version at first. And it's really, really, really confusing mystery. And actually, I'm going to sort of give it away. It's still a mystery today. And it's something that many scientists are still working on. And it's a fascinating question. So there's some really wacky theories as to why Earth ended up with more, for example, right-handed DNA molecules than left-handed. So one possibility is that the molecules that gave rise to life came from outer space. They came from meteorites that hit the Earth. And it's possible that those meteorites experienced what's called circularly polarized light. So you can imagine light that's traveling not in a straight line, but in a corkscrew pattern, sort of like DNA, same sort of pattern. And you can imagine that these patterns have handedness. The pattern on the left and the pattern on the right are actually different, and you can't turn one into the other. So imagine that for some reason, some meteor that was carrying the molecules of life encountered more of the left-hand side type of light than the right-hand side, or the other way around. So that would potentially favor chemical reactions that produced one-handedness over the other. But that's a really wacky theory that actually nobody knows whether this could possibly even happen, but this is an idea. So another possibility is that at the very beginning of life, there were an approximately similar number of, for example, right-handed and left-handed DNA molecules, but by chance, just by chance, there was slightly more of the right-handed ones. And over time, those right-handed ones made more and more of themselves until everything was right-handed. So that's a possible explanation. But the problem with that is you probably had many little areas on Earth where these kinds of processes were happening. Why did the same right-hand side win every time? Why isn't it about 50-50? And when you ask that question, you realize that we're sort of still back at square one, and we actually really don't know what's going on. But I think, we're working on it, and also you guys could potentially help. And the cool thing about this kind of question, this origin of life handedness question, is that you need a lot of different kinds of scientists to work on it. So for example, you need obviously biologists and chemists to understand some of the molecules and how they're interacting with each other and how they're building living things out of them. You need physicists and mathematicians to try to model what the biologists see in a way that you can actually make predictions about why the right-handed side won. You also need people like computer scientists, because this kind of science generates tons of data that needs to be processed. And you also need science writers and journalists, people who will actually publicize this kind of work so that more people are aware that this is a major question that has not been answered. And then, very interestingly, we also need astronomers, because there's a really strong possibility that some of the precursors to life came to Earth from outer space. And therefore, it really makes sense to try to understand some of the processes that happen in outer space that could lead to, for example, right-handed DNA winning over left-handed DNA. So there are so many different types of careers that you guys should really pursue if you're passionate about it, that you could potentially work on problems like this. So it's all in your hands. Thanks.
Thanks so much, Dan, for a great talk. As a chemist myself, I think about this handedness quite a lot myself. And so I have a couple questions here for you from the audience. One question that came up a lot is, we talked a lot about molecules being left-handed and right-handed. Is that also a reason why we ourselves are right-handed or left-handed or prefer a certain hand?
Yeah, so turns out that wherever handedness is concerned, people don't really know much. So it's not actually fully understood why some people are left-handed and some people are right-handed. It probably has something to do with events at the molecular level, but nobody knows the answer to that. It's another very interesting question.
All right, and another question is, when talking about the different theories about left-handed molecules and right-handed molecules and how the right-handed ones won, wouldn't the left-handed ones have also made more of themselves as well? And so how did the right hands win that race? Do you have any sorts of theories?
Yeah, I mean, so what, it's a very interesting question, and that's actually the central question that I was discussing is, so okay, in the beginning, maybe you had left-handed, right-handed versions, and even if they each tried to make more of themselves, why don't we have about 50-50? So it's possible that there were just happened to be slightly more right-handed DNA molecules, for example, in the beginning. And that small difference was such a powerful difference that over time, because they kept multiplying themselves, that even a slight unfairness in the beginning eventually led to an enormous advantage later on over billions of years. How exactly that happens and what would be necessary for that to happen is a mystery.
Mm. Yeah. So yeah, still a huge, huge mystery.
A huge mystery. Nobody knows the answer to this.
And so we talked a lot about the right-handedness winning, but do you know of any other organisms in which the left-handed hand won?
So in terms of DNA, no. All organisms use the right-handed DNA exclusively. However, there are other kinds of molecules, for example, amino acids, which are the building blocks of molecules called proteins. And there are actually some bacteria, et cetera, that have, where some of these molecules have an opposite handedness to the normal handedness. But even in those cases, most of their molecules have one preferred handedness. Sometimes they'll experiment a little bit with the other just to do something kind of cool. But generally, there's always a preferred handedness. And literally from flies to humans to centipedes to bacteria, everybody has the same handedness preference for their molecules, and nobody knows why.
Maybe there is a reason behind why a lot of us are more right-handed than others.
Yeah, I'm totally connected. Yeah.
But yeah, so that's all the questions I have for you today to answer here. There are some extra questions left over from the chat, and those will be answered in a video that everyone's going to get a link to later in your emails. So thanks, Stan, for answering those questions.
Yeah, sure.
Hey everyone, we're back with Victor. He just finished giving a wonderful talk earlier today about Huntington's disease and how it's been affecting his family and how he's working to learn more about the disease and how to cure it, and other Polycube diseases like SCA1, which he's been working with mostly in his beginning of his PhD. And so we just have a couple more questions for you that we'd love if you could answer. And so the first question that we have is, you mentioned a little bit about clumping. And does the amount of those clumps affect the impact of the disease?
Yes. So clumping, it's very controversial in the field. Some people think the clumps are good. Some think the clumps are bad. You know, in the olden times, we thought clumps, big clumps, cells die. Other people think the clumps are putting all the garbage, all the proteins in one area, so they don't affect other areas of the cell. So there's debate about the clumps. I use the clumps as an example because it's the easiest way to understand the disease, about how these clumps kill, or we think kill. But there are some scientists that are for and against clumps. So big clumps are bad, big clumps are good. The field is constantly changing with science when we get new discoveries. I personally think clumps are bad, but you know, we still need to know more about the disease and what it does. It's hard to know, you know, if you sequester all the protein in one area, you know, what's the normal protein? Because some of it folds correctly, but we don't know what that stuff's doing. So to answer your question, we don't know.
Okay, so following up then, going a little bit different direction. So that clumping action was because you have a bunch of cues in a row and the cues are dragging them together, we think. And those cues come from the CAG repeats that you talked about at the beginning. And you let us know that if you have too many CAG repeats, you can see this disease income. But what if you have too few? You put the healthy range at about 5 or 6. What if you have fewer than that number of CAG repeats?
Yeah, so for example, I could use mice. Mice have 2 normally. They have 2 of them. So they still have some. They don't have 0. And in any similar protein in this and any other organism that has it, they still have these cues. So there's no organism that doesn't have any cues. So the cues play some role. What role that is, we don't know, but they do play a role somewhere. So some scientists have tried getting rid of the cues using CRISPR, and the results vary depending on the lab. So it's still up in the air. If we just cut the cues out and not have disease, some say you survive, some say you die before you're born. I don't know how you get such big results from doing the same thing, but they do. So too few can be a problem, and too many can definitely be a problem. So you got to be in that healthy range. And the cues actually fluctuate between parents. When your father passes the disease on to the child, the cues can actually get longer. But when the mother passes it on, they can get smaller. So my grandfather died after my aunt because he passed on his disease, but it went bigger for her. So she had it earlier than him. So the cues they grow, they shrink. It's very, yeah. And then also, if you're at 37 cues, one cue above that 36 for Huntington's, you are in between. Like, you have the disease, so it's dominant, you have it, but you might not show it still. So it's like the 36 to 39 range of Huntington's. It's like an intermediate disease, smaller effect. You still have it, but not as deep as the other repeats. So there's work on that. The more repeats you have, the more severe the disease is.
Interesting. And so on that same, still thinking about, we're talking about the amount of cues. If we think about like the amount of diseased cells, does the amount of diseased cells also impact the severity of the disease in the patients? Okay,
so the disease cells, these are the neurons that are affected. So for each Polycube disease, a different brain region is affected. For Huntington's, it's the basal ganglia region, that region in the middle of the brain that had a hole in it in the picture. In SCA1, it's the cerebellum, which is in the back of your brain here. And so, each cell, they affect certain cell types. In Huntington's, it's the medium spiny striatal neurons. And in SCA1, it is the Purkinje cells. So over time, those cells will start dying and getting the disease. And so only those cells, though, are usually affected. So those are the disease cells, but there's other pathways that those cells talk to, and they connect to things, and those get affected. They don't die, but they're losing connections, or they're being interrupted. If you remember, every cell expresses these proteins for Huntington's and for SCA1, but not every cell is dying. So are they diseased? Are they not? How diseased are they? In SCA1, the cerebellum's affected, the back of the brain, but if you look at the cortex, these other areas of the brain, there's slight decay. There's slight effects there because it's expressed there as well. Why then only those cells there are dying? We're still working on that. We're still trying to figure out why certain areas, when it's expressed all throughout your body, why certain regions are more prone or more sensitive to it. Are they just more sensitive, or are they unprotected? Is there a protein expressed everywhere else in the body, but not in that region? And so everything's protected but that region? We're trying to discover that. And of course, to answer, go back to your question on the number, sure, the more cells that have and died, the more severe your disease will be. But it shouldn't be thinking of the number, it should think of the region and types of cells that are affected, really, and how we can stop that from happening there. If we stop the cells in the cerebellum from dying, that's great, but then maybe you live longer and now the cells in the cortex or other regions now start dying. Maybe it took longer for them to die. Maybe they were diseased, but at a slower rate, and they will die just slower.
So following off of that, for SCA1 or Huntington's or any other Polycube, do you still see that protein clumping even in the cells that you normally don't think of as being affected?
No, the protein clumping really is in those cells that are dying. And that's what led to people to believe that protein clumping led to cells dying. But you have all these other connectivity issues and other things happening before the cells die. Like I showed that timeline, you have at 5 weeks, they can't walk correctly, and then they have their thought process is affected earlier than the cells dying. So it's not that the clumps are killing the cells and that's the cause of the disease. Maybe the clumps are affecting different biological pathways that then lead to the cell dying, stuff like that. We're still trying to figure out, maybe they're clumping all the stuff together and some of it is escaping and being toxic, when you're trying to clump it all together to prevent the toxic function of it. Or maybe that clump kills it. Still trying to debate that, still trying to figure it out exactly. For SCA1, we know what it does normally, so we can more easily assume what's happening. But for Huntington's, we don't know what it does normally. So we don't know, we still can't, we can make a hypothesis, but we still don't know. It's very, very annoying. Trying to figure out what this protein does. It's weird to say that we don't know what a protein does normally, but we know when it's bad, we know what happens. You would just go, oh, it does the opposite, but no, it doesn't just do the opposite.
So you talked a lot about the impact of these cells dying, but when I think about a lot of cells in the body, like they can regenerate. Is that not the case for these diseases?
Correct. So neurons are very special in that once they've been made, they don't divide anymore. Your brain doesn't keep growing. Your brain is a certain size. And once you reach that maturity, it's done. The cells stop dividing. So if you lose a neuron, it's gone. So that's why you have to really take care of your brain. You can't damage it because that damage is permanent. You can't regrow it. So if you think of a simple A to B to C, one neuron, another, and another in a linear pathway, if B is gone, A can't go to C anymore in that straight pathway. So maybe it has to go around some other way, like we talked in memory a few weeks ago, how you get these memories by these neurons. If they're controlling your body and one neuron tells your arm to move and it's gone, well, your arm's not going to move anymore when your brain tells it to. It's going to move on its own kind of stuff. So sadly, these neurons cannot regenerate on their own. And that's why step 2 is so hard. How do you regenerate neurons? How to regenerate pathways? You have to take a lot of different drugs and other things to force those cells into a state that they can divide again. And getting that to happen is incredibly hard. We do it in the lab in cell culture, and so we force it to. And it's easier because it's on a little petri dish and we can do it in lab, and we just throw a lot of stuff at it to force it. But it's a lot harder in a living organism to make that work. And then how do you target just certain cells, certain very small cells in a certain small region of the brain to regrow, and not every other brain cell? Because your skull doesn't grow anymore. If you get your whole brain to double, that's going to be bad. So how do you target very small cells? Very challenging.
So this is a question that wasn't from the students, but I'm going to try to tee you up on it anyways. So Dan, at the end of his talk today, also brought up how the question that he's looking at is something that requires the efforts of scientists across a range of fields. Would you feel that that is the same for your study of SCA1 now or Huntington's in the future?
Entirely. There are many, many labs out there doing many great things in SCA1, in Huntington's. I'm currently qualifying for my PhD, and all these papers I'm reading are from many different labs all across America. And you read these things that they do, and so these other people out there influence what you do here. And then I'll maybe just come up with a discovery that I put out there, and other people will read it, or they'll talk to me, and we'll discuss together. My lab here at Yale is currently collaborating with 3 or 4 other labs right now on different projects. So within our university, we're collaborating. And then our labs also collaborate with other labs and other universities in Columbia, which is in New York, I believe. I think my Ivy League college is bad. I think it's in New York, and other in Minnesota, and we have a collaboration in San Diego. So we're collaborating very much because, like I said, you can't cure this on your own. You can't do it on your own. In 27 years since the discovery of the disease, we still haven't cured it. And that's with many people working on it. And so that's why I hope that one day these students here, maybe some of them will join and work with me on it. And it's going to take a long time. I expect in 20 years I'll still be working on it. And if by luck I cure one disease, I got 8 other ones to work on. And if I cure those 8, well, there's Alzheimer's, there's Parkinson's, there's many diseases out there. My work's never going to be done. There's always going to be something to discover.
Well, this has been fantastic, Victor. It was a great talk, and I think the students will appreciate having some more answers to their questions. And really, thanks so much for giving us this time today.
No, thank you for having me. I'm really excited to do this. It was a great honor for me to talk at your guys' event.
All right, so we're back with Dan, who gave us a really excellent talk today, talking about symmetry in nature and molecules and what happens when it's not there. So we're going to follow up with some of the questions that y'all asked just to make sure we get a chance to get to as many of them as possible. So Dan, first, you mentioned about the left-handedness and right-handedness of DNA, but is it possible that what if like artificial DNA was created to go in the opposite direction? Could that happen, and what would that mean?
Yeah, so that's a really good question. Thing is, nobody has found this DNA that faces the other way in any real organisms, but it has been created in the lab for the exact purpose of studying handedness in molecules. But no, there isn't any DNA like this in any living thing. So if the DNA was really the other way, it could work in very much the same way as long as all of the other molecules that DNA interacts with, including proteins, also had flipped handedness. So if everything is exactly flipped, it all works. But if you just put left-handed DNA into a right-handed environment, it's not going to work at all.
That's actually what I was thinking about as a follow-up question, because we talk about these molecular scissors a lot, right? And we know that the left-handed, right-handed scissors are sometimes difficult to use. I mean, that's kind of a silly analogy, but yeah.
Right,
so
no,
the entire system.
Mm-hmm.
Interesting. Yeah. And so on the topic of this left-handed versus right-handed and the entire system needing to be different, whenever you showed that picture about the sort of ocean with like left-handed, right-handed stuff, weren't the left-handed molecules also making more of themselves? And how did that happen?
Yeah, so that's one of the biggest problems with, the idea that there was a right-handed molecule that just made more of itself, because you ask, well, okay, but what about the left-handed molecule that made more of itself? So actually, there is no good answer. Nobody knows. That's one of the greatest mysteries in all of science. It's possible that if there was slightly more of, for example, the right-handed DNA than the left-handed, then over time, because it kept copying itself, it could have overpowered the left, so much that there's basically no left-handed DNA left anymore. But how that happened and whether that happened is a complete mystery.
Do you think it has to happen with like interactions with other molecules as well? Like sort of by chance, other molecules that interact with it also were right-handed, and then that sort of just influenced those to be propagated?
Yeah, so that's possible. It's possible that there was a small imbalance in, slightly more right-handed than left-handed DNA, but also the molecules that are needed to, help copy the DNA and make more DNA also had the same sort of unevenness. And, it started from a small amount, but if you imagine that every time DNA copies itself, it multiplies itself by 2 times 2 times 2 times 2, and every time you get exponentially more DNA. So even a small difference in right-handed versus left-handed populations on the early Earth could very quickly turn into like 99.999999% right-handed.
Right. That makes sense. And so we are talking about DNA from like an early stage, and then we're sort of jumping to humans because that's what's most relevant to us a lot of times. But does this hold true for other organisms in addition to humans and animals about the handedness of DNA?
Yeah, so it actually does. The DNA is right-handed in every animal, but also in every plant, in every fungus, in every bacterium. Anything that has been discovered, any species of organism that has been discovered ever, has right-handed DNA and right-handed DNA only, which is a really big mystery. But it also, really shows how really all life on Earth is interrelated. And also it supports the idea that all life on Earth came from one common ancestor that had right-handed DNA.
I see.
Yeah.
Awesome. And are there any inherent differences between sort of the right-handed and left-handed, or are they, as far as people understand, basically the same? They're just flips.
Yeah. So the difference is only in the right-handed or left-handed DNA's interaction with other right-handed or left-handed molecules. So if you take everything right-handed and just flip it to being left-handed, it would be exactly the same. But if you only flip the DNA but didn't flip the protein, for example, then it would be like trying to shake your own hand, and it would be awkward, and not all of biology, it would break down. So it's important for things to match, but once they match, it doesn't matter if it's right or left.
I see. Awesome. Cool. This stuff is really fascinating, and I struggle with some of these. A lot of the pictures that you showed about whether or not they actually line up on each other, I have a hard time thinking about that. But it's really awesome to see people studying this sort of stuff and trying to understand what it all means and make it all make sense.
Yeah, the 3D part can be a little confusing, especially on a PowerPoint. It's it's easier to show this to yourself, this handedness using actual models, but unfortunately that is impossible with Zoom. So
yeah, well, it seems like the hand example is really good, though. I think that's really visceral for people.
Yeah, that's probably the best example is actually on our own bodies. Yeah.
Great, awesome. Well, thanks a lot, Dan. I really appreciate it, and we'll check you next time.
All right, yeah, no problem.